Dr. Gorevic joining us for a discussion on treatment strategies for amyloid disease. Dr. Gorevic is a professor in the Department of Rheumatology here at Mount Sinai. He obtained his medical degree from New York University, where he stayed on to complete his residency in internal medicine, as well as fellowships in rheumatology, as well as allergy and immunology. He later served as chief of the Division of Allergy, Rheumatology, and Clinical Rheumatology at the State University of New York at Stony Brook for 10 years. Dr. Gorevic has been a faculty member at Mount Sinai since 1997 and has served as division head and Stratton chair of the Division of Rheumatology for 15 years. His main research interests are amyloidosis, cryopathies, and autoinflammatory diseases. His work in amyloidosis has received grant support from the VA, NIH, American Heart Association, American Federation of Aging Research, and the Alzheimer's Foundation. Please join me in welcoming Dr. Gorevic.
Okay, I have to make sure you can hear me in the back row, and then I guess we get going. This is an interesting conspiracy here. It's always interesting to think about what would happen if two people met. If any of you have seen Steve Martin's play about Picasso and Einstein at a still existing bar in Montmartre, and what they would have said to each other. The term amyloid was first really coined by a fellow named Rudolf Virchow. He was very young when he coined the term, and he wrote a book. And the book was probably written when he was in his 30s, and it was called Cellular Pathology. And he described this entity called amyloid. It had actually been described years before that, but he's the one who used the word amyloid, which is confusing because there's no starch that's really significant, although we'll talk about that. But he also was a thinker, and he made a lot of seminal observations. One of which is that he thought that this condition called amyloid, and he was clearly looking at secondary amyloidosis, which we'll talk about as we get further with this, is that he thought it was a plasma cell dyscrasia. He thought that this was something wrong in the blood of these patients, and that's what was causing this stuff to deposit. Now, those of you who know about Virchow know that he lived for a long time. He lived on until about 1901. And he did lots of interesting things. He was a pathologist, but he also saw patients. He went into politics. He went on archaeologic digs with Schliemann. And he did all sorts of different things, including a journal which still exists to this day that has his name on it. And he died in 1901 in a thoroughly modern way. He fell off one of the newer electrified trams, fractured his femur, and died of the complications of that. Now, also in 1901, there was a Prussian psychiatrist by the name of Alzheimer, and he was studying a patient named Auguste Deterre. And this was the initial description of the disease that he eventually wound up looking at pathologically after she died. The remains were revisited in 1988, and in fact, she did have Alzheimer's disease. And he described it, and about 10 years ago, later than that, it got his name on it. He didn't know very much about amyloid, but amyloid was a very important part of this. Now, let's see if I can move this. So I'm trying to move this forward, and I think I just turned it off, so I need somebody's help to start this up. Okay, now to go forward, which one? Got it, okay, thank you. All right, secondary amyloidosis. This is a model for secondary amyloidosis. What we did, it was discovered in 1920, and a fellow named Smetna discovered that if you overdose mice with cheese, you'll get them to make amyloid. The amyloid will develop in the spleen first, then in the liver, then in the kidney. This is the liver. And you can see this fibrous material here with a cell next to it, which may be very busily trying to engulf it. And this turns out to be amyloid. But the other thing that you might recognize is that close by is a blood vessel, and in the wall of the blood vessel, there's more amyloid. This is a plaque in the patient who has Alzheimer's disease. And what you see in the plaque is that there is an amyloid core, which is stained birefringence with Congo red. And around this are dystrophic neuroids and neurofibrillary tangles And we know that the composition of the amyloid is a protein called A beta, and the neurofibrillary tangles is hyperphosphorylated tau. But again, if you look closely, there's blood vessels. And so congophilic angiopathy is a common theme with all of this. So the objectives today are the spectrum of amyloid diseases in 2017, proteins and genes, amyloid on the medical service. I'm going to give you 3 cases from a place called Mount Sinai Hospital, which we've seen over the years, and an evolving landscape of treatment and prevention options. I first talked about this shortly after I came to Mount Sinai in 1997, except at that time, Dr. Kola was facing that way instead of this way. These are my disclosures. This is the number of publications we planned. It's estimated we're going to be up to about 50,000 publications. And I put this here because that's about when I started to work on this subject. So what is amyloid? Amyloid consists of a number of different things, but the major component of amyloid is the fibril, which you can see under electron microscopy. The fibril gives it its specific characteristic, which is apple green birefringence when you add on a dye called Congo red. This is a water extract of fibrils, which goes back a long way for me. It was from a patient who had familial Mediterranean fever, whose spleen we were studying, and we were doing the sequence of a protein called AA, which we isolated from the spleen of that patient. This is a solution of Congo red. It's actually sort of orangish. It binds to the fibrils if you spin them down and turns dark red. That's a characteristic called metachromasia. All forms of amyloid have a second component called P component, which is a pentraxin, which you can visualize in purified preparations. And Virchow was correct to the point that there are glycosaminoglycans in every form of this. So the definition of amyloid, to start off then, is homogeneous, maybe not so hyaline, mostly extracellular material with typical fibrils by EM, metachromasia, Congo red birefringence, binding to specific dyes, one of which is called thioflavin T. Thioflavin T is now licensed as a reagent for imaging amyloid in vivo. And in fact, if you go over to the imaging center, you will see that it's being used for CNS amyloid, but it also binds to systemic forms of amyloid. But under this rubrum are multiple chemical types. We're up to 34 amyloid types as of 2017. We have a nomenclature committee which every couple of years updates this. We're not going to talk about all of these, but all of these proteins have been shown to form amyloid in association with different clinical diseases. The ones we're going to focus on are the apolipoproteins. There are now 6 different apolipoproteins which are known to form amyloid and give different clinical syndromes. The main one we're going to talk about is serum amyloid A, the immunoglobulin gene family, which is immunoglobulin light chain, occasionally a heavy chain, and beta-2 microglobulin. Beta-2 microglobulin is the one that causes what we call dialysis amyloidosis, but this is AL amyloid. And the third one we're going to talk about is transthyretin, which is a transport protein which causes amyloid. There are a number of cerebrovascular amyloids, and it's important to remember them, such as Alzheimer's disease, but it doesn't stop with Alzheimer's disease. It stops with other variants of this. So what do we see in a referral center? We see systemic amyloidoses. We'll talk about AL, we'll talk about AA and ATTR. I'm not going to talk about beta-2, and there are a number of other heredofamilials. These are generally rare diseases. Alzheimer's disease is not a rare disease. This is a much bigger nut here. But we also see a number of others that are seen usually by specialists here. These are laryngeal amyloids, ocular amyloids, genitourinary amyloids, pulmonary amyloids, and it goes on. And there probably are age-related forms of localized amyloid which are extremely interesting to us from a variety of different points of view. So all of these diseases have something in common. What we're talking about is the fact that proteins can take on multiple different conformations, and sometimes these conformations lead to normal folding intermediates that you would expect, but that it can go to an off pathway or a number of different off pathways. And then what you see are a different number of aggregates. These aggregates may be amorphous aggregates. They may be the amyloid fibril, but they may also be oligomers, which are multimers of the monomers of these particular proteins. And the questions are then, what causes the protein misfolding that is relevant here? And there are a number of factors. We're going to concentrate a lot on mutations as we go along. And you should be aware that amyloid is really part of a spectrum of diseases, which includes things like Parkinson's and Huntington's disease, where there's also aggregate issues, and including the preanoses, which we're not going to talk about today, but are very much related in terms of the thought process. Now, in all of these cases, in all of the diseases that we're talking about, the protein is synthesized in a cell, usually a single cell, and there is the normal factors that lead to the export from the cell. There may be a mutation which is apparent on the intracellular processing, and then there are a number of things that will occur in the extracellular compartment, including the normal function, obviously, and the extracellular influences. But as you go in the process that leads to amyloid, the sequencing is that you will have these oligomers or protofibrils, and the oligomers have a number of functional consequences which have been extremely interesting in terms of thinking about these diseases. And it's only later that you add on the glycosaminoglycans, which facilitate the fibril formation, and the serum amyloid P component, which has a very integrated relationship to it. Regardless of what type of protein we're talking about, these are proteins that adopt what's called a predominantly beta-pleated sheet configuration. This is transthyretin. This is an immunoglobulin light chain. Has a very characteristic pattern, which we use for the definition of these fibrils. So I keep a tabulation of mutations associated with different types of amyloid, which I update every 6 months for up to date. So if you want to see the most recent one, just look under genetics of amyloid, the card on up to date. But there are a number of mutations which are important in terms of the hereditable and acquired forms of amyloid. The ones we're going to talk about specifically are TTR, of which there are now 130 different mutations which have been described. And I will also talk a little bit about Pyron. Pyron is a protein which is associated with amyloid. It's not part of the amyloid, but which may have an important modulating effect. And there are 174 coding mutations that are associated with specific phenotypes that we'll talk about as we go on. So what is it that we see in a referral center? We are one of probably 4 or 5 major referral centers for amyloid diseases in the country. We see the systemic amyloids, light chain amyloid, about 18%. This is a rare disease, maybe 15,000 patients in the United States and Europe. Secondary amyloid, it's changing. We're seeing more and more of it recently. I'm not quite sure why. About 7%, but relatively rare in Europe and North America. And then TTR, amyloid due to transthyretin, which can either be due to a mutant molecule, and there are about 5 to 10 cases, 10,000 cases of familial amyloid polyneuropathy. Then we'll talk very specifically about a particular mutation called isoleucine 122, which is a very important public health issue. In addition to that, there's wild-type TTR, which means that as far as we can tell, there's no mutation. And this is about 7% of what's seen in major referral centers, such as the UK Amyloid Center, probably as close to what we're seeing here. And this is much bigger because we're talking about a significant number of patients that are affected by this. So let's start off with a case. And I said the case is from Mount Sinai. This is a case from Mount Sinai. This was a patient who was on our service a number of years ago. She was referred to me because I'm a rheumatologist, that's my day job, and I see these patients. 54-year-old woman, tightening of the skin, decreased oral aperture, she was xerostomach, she had dysphagia, and she was originally diagnosed as scleroderma. which made a great deal of sense. She was actually treated for scleroderma, but she was referred to a scleroderma center. At that time, there was one in New Jersey, and the person who saw her there realized that there's some things that just didn't quite fit. She didn't have a history of Raynaud's. We in rheumatology get very suspicious if our scleroderma patients don't have Raynaud's. She had a large tongue. This is a picture Dr. Wu took here at Sinai, and this is a big tongue, and it turned out there was a lot of amyloid in it. She had jaw claudication. Jaw claudication is something that we in rheumatology associate with giant cell arteritis, but in this case it wasn't giant cell arteritis. And she then had a few other things that didn't quite fit. She had a cardiomyopathy, she had atrial arrhythmias, and so the person who saw her did a deep skin and muscle biopsy. So here's the skin and muscle biopsy. You see this amorphous material below the epidermis, that's amyloid. You see this material in the wall of blood vessels, that's amyloid. And this also around the agnexa, which is the hair follicles and stuff. This is the type of amyloid, when it occurs in the skin, is a tip-off that you're dealing with a systemic problem rather than a localized problem, because we do see localized amyloid in the skin. And in fact, she was seronegative. We do capillaroscopy in rheumatology, and she didn't have the typical pattern. She had a large liver. She was spilling a lot of protein. She had hypoglobulinemia, and this was her skull. So you see, we're talking about something very different here. She had 45% of her serum and 70% of her urine was lambda, a lambda immunoglobulin light chain. And when she had a bone marrow done, 40% of the cells were plasma cells. So this is an example of multiple myeloma presenting as amyloid. So what did we do? This is the Bence-Jones protein in the urine. It was a lambda Bence-Jones protein. We isolated it and we sequenced it. It's hard to isolate lambda and sequence lambdas because they usually have a blocked end terminal, so that's why we got residues 3 to 21. And then we took the actual biopsy, extracted the amyloid out of the biopsy, and looked by mass spectrometry to see if there were any informative peptides. And there was one peptide that was interesting because that peptide matched this peptide. And what we were looking at was the sequence of an immunoglobulin light chain, a lambda immunoglobulin light chain at the end terminus, the variable part. And then we did the next thing, which was to take a slide of the myeloma. This was the bone marrow biopsy, and PCR out the light chain sequence of using those particular sequences. And it turned out that what we were looking at was a lambda 3R, which is a specific gene in lambda. And it was the same sequence that we found in the myeloma cells as we isolated from the amyloid. So I'm drawing the circle between the light chain and the amyloid. And there are a number of data about this. This particular subgroup or particular gene of lambda is one that's interesting. This is the sequence of a variable region. There are frameworks, and then there are the hypervariable regions, which are where the antigen binding sites are. And you can see that in this particular patient, there were some mutations that were there. Some of them were expected because they were mutational hotspots. But we also know that some of these mutations probably profoundly influence the structure of the lambda. And this has been done by doing mutagenesis experiments looking at the sequence of different lambda 3Rs. And those very specific sequences are very much related to the process that leads to fibril formation. So what we're talking about is a type of amyloid that's due to immunoglobulin light chain. The plasma cell is making a light chain, but the amyloid itself is coming from the variable part. The constant region has been really digested out, more or less. And here is the hypervariable loops and the frameworks. And what we're saying is that there are changes in the sequence, specific changes in the sequence that you can relate to the fibril. That's very important because as you start to think about how you might interfere with this, you could use those epitopes or you could use those sequences to try to design peptides that might interfere with the process. This is from a very recent paper that just was published in Blood from the Mayo Clinic, where they're looking at this lambda 3R, both in systemic amyloidoses and also in the localized forms of amyloidosis, which I sort of indicated, many of them turn out to be AL also. So this is a light chain variable region gene which is strongly associated with amyloid, and there are about 3 or 4 of them that account for most of the lambda sequences that we see, and most of the AL amyloids are lambda rather than kappa. Systemic amyloid AL is a devastating disease. It presents to every service here. You had a very excellent grand rounds on this that was given by Karen Osman about 4 or 5 years ago. And I think Karen went over a lot of this, but this is a very important type of amyloid. It can involve the kidneys to cause proteinuria, it can involve the heart, it can involve the liver, it can involve and all of these patients come in looking quite sick. This is purpura, and this is macroglossia, this is carpal tunnel syndrome. So it can present to any one of our services, and it usually does. And as of about 2 weeks ago, we had a patient with cardiac amyloid who was on the cardiology service, again with AL amyloid. Lousy prognosis, but getting better. The prognosis this is the database from the Mayo Clinic. In the 1930s, most of these patients were gone within about 5 months. That's very different now, and it has gotten progressively better. And by 2006, the number was up to 47 months. And this is really a function of bone marrow transplantation, which has been a really important therapeutic tool. But there are patients who will not benefit from transplant. And these are the patients who have cardiac amyloid. They still have abysmal prognosis. The AL patients who have AL, who have this particular congestive heart failure, have a very compromised. And so this is a major challenge. So in terms of thinking about treatment, because we're focusing on treatment, AL amyloid, myeloma leads the way. We follow from our myeloma colleagues, and how are we going to do this? And basically what we're talking about are these proliferating plasma cells. We're making increased levels of free light chains, and there are a number of different approaches to this, including some new ones which are spilling over to amyloid, and then autologous bone marrow transplantation, which has really changed the landscape, but is only suitable for maybe 25 to 30%. But where are we going? And where are we going is that up to now, in the treatment of this disease, the main thing has been to knock down the precursor, get rid of the light chain. You can do it on a transplant, you can do it with all these poisons, but there are people who are starting to think about whether there might be a different way of doing it. And the way of doing it is what we call the Holy Grail. The Holy Grail is, can we develop an antibody, which is a conformational antibody, which will react specifically with those light chains which are forming the amyloid, and which are showing epitopes that may only be exposed in the amyloid or in the oligomers that lead up to the amyloid. And so they made a humanized murine monoclonal to an AL cryptic epitope, and it has been done in phase 2 studies that were published last year. And here you're looking at cardiac evaluable patients, looking at the NT-proBNP, which is a measure of cardiac dysfunction in this group, and showing that the patients who got repetitive infusions with this antibody, an antibody which they claim reacts with the amyloid and leads to clearance, so very different than knocking down the precursor, or which reacts with soluble aggregates, which would be these oligomers which are in the blood. And they have gone through a number of different drills to try to show this, and there are 2 very large multi-center international studies which are now looking at this as a treatment for naive AL with cardiac disease, and then previously treated AL with persistent cardiac dysfunction. A very different approach. Case number 2, amyloid at the interface of inflammatory bowel and autoinflammatory disease. We still follow this woman. She's 71 years old. She was found to have AA amyloid on an endomyocardial biopsy. I was not able to get the actual pictures because Dr. Beasley emailed it to me last night. So I'm going to use something different, which is that we used to look at these by basically looking at them on 2D gels. And what we would do is we would look at endomyocardial biopsies, or we would look at histology if we had material that we could look at. and we would analyze the proteins that you would see in the amyloid. And in this particular type of amyloid, the major protein is a protein called AA. As opposed to the light chain type of amyloid, this is a heart of a patient who had AL amyloid. You see the intact light chain and the fragments that we talked about. She had a history of fevers. She had a history of pericarditis. The cardiac MRI showed severe left ventricular systolic dysfunction and hypertrophy. A cath showed non-obstructive coronary artery disease. There was no history of hypertension. She was not orthostatic. And I'll say that we are now quite a number of years later, 2017 from 2009, and she's remained stage 2 heart disease, which is relatively well compensated. Now, Crohn's disease was diagnosed in this patient by Dr. Present here at Mount Sinai. So I believe that diagnosis. And she had a lot of issues with Crohn's disease. She had small bowel fistula. She has sclerosing cholangitis, and she remains on ursodiol, which may or may not be significant. And her IBD had been treated with various things, including TNF antagonists. And recent colonoscopies have shown minimal Crohn's, but also congophilic angiopathy. She's got the amyloid in her bowel. She probably has it, she almost certainly has it in her kidney too. When I first saw her, initially, her SED rates were very high. She had a lot of acute phase reactants. And a protein called SAA, which you can measure, was 158.7. Normal is less than 10. This is a huge increase in this particular protein, which occurs when you have a very inflammatory issue. We've been able to knock it down with Humira, but it doesn't work very well. She had urinary protein. That has gotten worse. She's now on dialysis and waiting for a kidney transplant, which raises a lot of issues. Now, incidentally, we found that she has a mutation in a gene called MEFV, which is Pyron, and that she's heterozygous for the M694 pathogenic variant. There is no family history of amyloid, IBD, or periodic fevers. This is not a person from the Mediterranean. Her family comes from Scotland, as she was born in the West Indies. So, this particular type of amyloid is AA, and it comes from a protein in the blood called SAA, which is a high-density lipoprotein, acute phase reactant, every bit as good as C-reactive protein in terms of what it does. It undergoes cleavage, like a lot of these proteins, and then it forms amyloid. And the AA amyloid that you see can complicate connective tissue diseases, can complicate chronic inflammatory diseases, and I draw your attention to two things here. One is familial Mediterranean fever, and the other is Crohn's disease. This disease is changing. When Virchow saw it, it was the predominant form of amyloid that they would see. Autopsy series from certain parts of the world continue to show a significant incidence of the secondary amyloid. But autopsy series from the West report a progressive decrease. There is a varying incidence between populations. In Turkey, Syria, Egypt, and Israel, FMF, familial Mediterranean fever, is a common cause of AA and is responsible for a significant percentage of renal disease caused by amyloidosis. This is an update of a paper that I published with Dan Bunker, who is now junior faculty at Georgetown, was a fellow in our division. It's an update that basically shows you what we're looking at. We saw about a lot of Crohn's, but that's because of Mount Sinai. We also saw IBD coming in through various other routes into this, and we also saw some of these patients had Pyron mutations. So what's the Mount Sinai connection? This is the Mount Sinai connection. You may not realize it, but the disease that we call familial Mediterranean fever was originally described from Mount Sinai Hospital. And it was described by Shepard Siegel, and he described a group of patients which he published, which he called familial paroxysmal polyserositis. And this is Crohn's disease. And it's interesting to sort of think about these two things. These pictures come from Barbara Niss in the archive, so I'm very proud to have some of that stuff to look at. In the days before the medical school, a lot of our physicians came from Columbia. And so did Beryl Cohn come from Columbia, and so did Shepard Siegel. This is Shepard Siegel a house staff photograph on the top of one of our buildings in 1935. And I show this picture because his son, Fred Siegel, who was head of immunology here several years, eons ago, told me that his dad definitely had the same disease. He was he actually reported on himself. Now, Burrell Crone described the disease that we now call Crohn's disease about the same time that Shepard Siegel was going through his house staff training here at Mount Sinai. And this is an example of something that you think about is, what would happen if these guys had met in the faculty dining room, and would they have talked about this? Because it's very interesting. The main description of amyloidosis complicating inflammatory bowel disease includes a number of people that I'm sure you recognize, because the really big series on this was a 50-year experience with 25 patients. So there is this interesting overlap here. What is Pyron? It's a gene. There are now 307 sequence variations. I said 174 of them. There's a database. The large majority of them cluster in exon 10, including the 694, which is the one we're talking about. It's primarily expressed in polymorphonucleoleucocytes and mononuclear cells. Before colchicine, the incidence of amyloidosis among Sephardic Jews was as high as 30%. If you went to, in the population surveys that were done, and they surveyed Moroccan Jews, 1 out of every 5 of them carried an MEFV mutation. Some mutations are more, so including the 694, are more associated with disease severity and amyloidosis across populations, except we're also seeing E148Q, which is a low penetrance variant that's in exon 2. And it's been proposed that the Pyron inflammasome evolved as an innate immune sensor to detect bacterial-induced modifications of RhoA. So it may have had actually a protective function. It's like, why do people have sickle cell? Why should so many people be carrying around these genes? And we're in the process of doing a study with the help of the people in our Personalized Medicine Institute, looking at the biobank and trying to understand these mutations, because Shepard Siegel described 50 patients. 38% of his patients, including himself, were Ashkenazi Jews. There were only 4 Sephardic Jews in that population. There were a lot of Turks, Maltese, and only 1 had amyloid. But here is our data. This is Mark Matza, who is a house staff here, soon to become a rheumatology fellow at Mass General. Dan Bunker and myself, 60 patients because we started sequencing this gene when I arrived here in 1997, which was when the gene was first described. So in our 60 patients who have the mutations, we're seeing a lot more Sephardic Jews. And it's been very interesting thinking about the derivation of these Sephardic Jews, because one thing I've learned is that a Sephardic Jew is not a Sephardic Jew is not a Sephardic Jew. There are different subpopulations, and it's interesting to think about how these mutations fit in here. But we're also seeing the whole potpourri of people who live in New York. We're also recognizing, now that we have these mutations, that we are seeing atypical phenotypes. Not all of them have these periodic fevers. Some of them just have fever. Some of them have chronic, have a disease that looks like Behçet's disease, or a disease that looks like Sill's disease. And chronic GI, I think, is an area that needs to be looked at a little bit more. This is our experience. We also see a lot of heterozygous people who have these diseases, and that was not supposed to happen because we thought initially it was a recessive disease. It's not. And then again, we see amyloid as a phenotype. So how do we treat secondary amyloid? We treat the underlying infection. We reduce the source of inflammation. We remove the tumor if it's caused by a tumor. We reduce the level of SAA. This is really critical. You have to get the precursor down. And that's one of the things we do with some of these anti-cytokines. Initially, we thought it was anti-TNF, but the answer is it's not anti-TNF. We're getting much, much better results with anti-IL-1 and with anti-IL-6. So amyloid and inflammation has a very important cross issue. We're talking about inflammasomes that aggregate in or form multi-protein complexes in response to a number of different stimuli, and which lead in turn to the generation of IL-1 an IL-18, an S100, some specific S100s. And what we know is that some of these diseases, the inflammasome may be important pathogenically. I don't know anybody who's looked specifically at the GI tract for the inflammasome issues, but given the current interest in the microbiome for inflammatory bowel disease, it's obviously something which should be done. But also some of these crystals, some of these proteins can form amyloid. Now, secondary amyloid we have wonderful models for, and a very enterprising gentleman in UK, Mark Pepys, did a wonderful experiment where he actually looked at the P component, which has been his life's work. And he made an interesting observation. P component circulates in the blood, but it also associates with the amyloid. It does this in the experimental models. So he thought, I'm going to get rid of the P component. I'm going to develop a molecule which will deplete the P component. Didn't work. Although they're still looking at it now in Alzheimer's because it may have a function in the central nervous system. But he found he could deplete it by about 80% in the blood. And then he got the bright idea of making antibodies to P component. And lo and behold, he was able to completely eliminate the amyloid in the animal model. And this is what was published in the New England Journal about late last year, in which they're using depletion followed by an antibody to P component and showing improvement in the tissue amyloid, at least using very general types of measures. Third patient, a referral to Mount Sinai Heart. 61-year-old Nigerian man, first seen in 2005, progressive dyspnea, dilated heart, echocardiogram showed ground glass opinion, a large aortic aneurysm, no evidence of this. This is his bone scan. If you ever want to make a diagnosis of amyloid in the heart, in this particular type of amyloid, do a bone scan. And this is his workup. Acute phase reactants are normal, no Pyron mutations, no indication of anything. And unfortunately, he came back here a few years later and died. But he had 6 years that he was on no therapy between the time he was first seen here. And I want you to focus on the word Nigerian, because we do get referrals from all over the world to Mount Sinai Heart. And this man flew from Lagos to be treated. And when we evaluated him, we found that he had a mutation. And the mutation was at position 122 of a protein called transthyretin. It turns out that he is homozygous for this. And this mutation is what we call ILU-122. We can do this much better now, and we do this routinely on the endomyocardial biopsies. It is being sent to the Mayo Clinic, although we're in the process of trying to set up the methodology here. And this is mass spectroscopy, where we look at the amyloid, we isolate it, or it gets isolated, and then it's put through mass spectroscopy and sequenced. And you can do that on a tiny, tiny nodule of amyloid in the heart. This patient has TTR with a particular mutation. This mutation is one of 130 mutations that occur in TTR. We're not going to bother you with all the other mutations. The two that are interesting here is isoleucine 122, and I'll mention 119 very briefly. The readout in these patients is heart and nervous system. When you look at all the different mutations, they can present with predominantly heart, or they can present with predominantly nerve. And we see patients with both types here that get referred for treatment. The isoleucine 122 is I'm particularly fond of this because I first described this in 1988. It was a patient I was following at a VA hospital on Long Island. I thought the patient had AL. But it wasn't until after he died that we were able to work it out and show that he had this isoleucine. This turns out to be a cause of late onset cardiomyopathy. Late onset cardiomyopathy we see in a variety of different mutations. There are two mutations that are important in the New York area. We call one the Italian mutation, the other one the Irish, because everybody has either an Irish or an Italian name. But this mutation is a significant polymorphism in the African American population. 3.9% of patients are carriers of this particular polymorphism. It's also prevalent in the Afro-Caribbean population. We've had more than one patient that's come from Trinidad or from Barbados who has the same issue with cardiac amyloid. And there are 1.3 million people who carry this allele, including 13,000. And it may account for a significant percentage of the increased incidence of cardiac disease that is not completely diagnosed in the African American population. Very interestingly, a huge male predominance. And this is a study which was just published from West Africa, and you can see that the mutations are all there, as you would have expected. And in some of the West African countries, including Nigeria, they're really quite high. So what we're looking at is something that is circling around. Well, what do we do about it? Well, these mutations are all destabilizing. When you have the mutations, you go from the tetramer of prealbumin or transthyretin to a monomer. And they destabilize and they force you to the monomer, and the monomer forms these aggregates. So one of the treatment protocols has been to stabilize the tetramer. And there are a number of molecules which are being looked at. The one that was first out of the bag was one called diflunisal. Somebody said to me, do you know what my diflunisal says? I said, sure, I've been using it for about 40 years. It's not a terribly good NSAID, but we do use it. It's relatively easy on the stomach. It actually stabilizes the tetramer. There is a molecule which is available in Europe and about 4 or 5 other countries called tafamidis that also stabilizes here. And then there's a lot of interesting drug discovery, including rediscovery of a Parkinson's drug called tolcapone, which also apparently works this way. So this is one approach, is to prevent the monomer from forming the aggregates. And most of our patients are now on diflunisal. Many of them are going to be on tafamidis. But there are a number of other therapeutic options which are available to us, including a number of bivalent ligands, cyclodextrans. I'll talk a little bit about antibodies to cryptic epitopes, and then a few things which come out of the nutraceutical world, such as would you use curcumin? And would you use green tea? And the answer is yeah, you would. And there are studies where people are looking at it because this particular compound suppresses the tetramer, dissociates at a site distinct from T4, and disrupts mature TTR fibrils. And curcumin induces oligomerization to a non-toxic off pathway. And then here's one of our favorite antibiotics, because we in rheumatology use it for rheumatoid arthritis. We use it for all sorts of different things. But among other things, it disrupts fibril formation and inhibits MMP1. And I mentioned that my patient was on URSO, and we're actually putting a lot of these patients on URSO based on this. So TTR amyloid has undergone a major progression over the last 15 years. We started off in the early 1990s with liver transplants, and then we moved on to other things. Amyloid P antibodies started to begin used, and there's the proposed nutraceuticals. The transplants have fallen off, and it was a major part of our transplant program. One of the reasons I came to Mount Sinai was because of your bone marrow transplant and your liver transplant programs. And the most interesting thing that's going on now, almost ready to be released, is the use of a whole new different class of compounds to inhibit the production of the precursor. So here we're not talking about anything that works on the protein. We're talking about inhibiting the mRNA or inhibiting or silencing the RNA. And there are two different approaches which dot the New England Journal these days. One is using these antisense oligonucleotides, which degrade mRNA in the nucleus, or to use the silencing RNA. This is the generation of silencing RNA. And there are two pharma companies, one on the West Coast, one on the East Coast, and that one have been using antisense oligonucleotides to TTR to knock it down. You can make a human knockout, and they seem to do extremely well, which is nice to know because we were not really sure that that was going to happen. They're about to release their phase 2-3 studies. Some of the phase 1 and 2 have been, so we expect that this will be, we are participating in these studies, so we are participating in all of these studies. I mentioned that in my disclosures at the beginning, but I'm not the lead investigator. We will find out. So strategies for the treatment of amyloid. And here we're going general. We can use green tea. That's been used for TTR, A-beta, and a couple of others. We can use curcumin, again, A-beta. This is Alzheimer tetracycline, again, a TTR, beta-2 microglobulin. We can use trans-suppression. There are mutations that are protective, and there are two mutations that are interesting. One is a mutation of TTR at 119, which I referenced before, And then there's a mutation in the APP, or Alzheimer precursor protein, at 673, and the people who are affected by these mutations have a much more moderate form of disease. So it's thought that these may be protective. And there also, it turns out in a big study that was done in Copenhagen and Denmark, this may actually lead to a longer lifespan and better cardiovascular lifespan. Absorbent columns for beta-2 microglobulin, this is the dialysis amyloid. Peptide inhibitors of adhesive segments are being looked at. A lot of interest in camelid nanobodies to try to get to the specific epitopes that are important in aggregation or to block proteolysis. Protective chaperones. Chaperones seem to be very important. Some of the chaperones are very important. There's one called Bricos, which was originally described in a particular type of neurodegeneration called ABRE, because it was described in Britain. And this is being looked at as a generic therapeutic. Antiproteases have a sort of a mixed history here. It's been very popular in Alzheimer's, but really hasn't gone very far. And then there are peptidic inhibitors of HSPG, heparan sulfate binding. We actually participated in a study that looked at that a few years ago for AA amyloid. And then immunotherapy for amyloid. We talked a bit about this. There are vaccination strategies, and then there are passive immunotherapy. Vaccination therapies have been mostly used for Alzheimer's, and they include fibril aggregates, oligomer-specific subunit proteins. This is the original Alzheimer vaccine, which had a really checkered history because it caused a lot of inflammation, but actually depleted the plaques. So this is still an area of active investigation, including A beta and late tau. Late tau is later in the evolution of this. Passive immunotherapy, it's really a huge industry with regard to A beta, a number of epitope-specific antibodies, bispecific antibodies, antibodies. Mis-TTR is to a specific epitope in TTR that gets exposed when it becomes unfolded, and people are using that as a way, and also gamma globulin. Gamma globulin has been used, IVIG has been used for A beta and AL. The A beta studies were done at Cornell. So summing up, where are we with regard to treatment strategies for amyloid? We would like very much to identify the risk factors and screen for early deposition. This is a huge industry. These patients with Down syndrome, because they have 3 copies of A beta, APP, they're at a huge risk to develop Alzheimer's. Yet you have the facility to try to get at it early. What we're doing now is looking at the progeny of our patients to try to understand when these diseases start and how these therapies might work, because we've been so much focused on deposition phase and now only getting to clearance. So you can identify risk factors, you can screen for early deposition, you can stabilize the precursors, you can have therapy targeted to the toxic prefibrillar intermediates. There are a number of other things that have been looked at, inhibitors that prevent deposition, disrupt the fibrils. I've given you examples of that. We talk about sand in the sheets, which are these little molecules that go into the beta sheets and disrupt them from forming the fibrils. Chaperone-directed therapies, anti-inflammatories. I didn't really go into that in much detail, but it is an interest, certainly for neuroinflammation, still an interest. Are there ways, and I was thinking about Dr. Murad's grand rounds a few weeks ago, are there ways that you might enhance macrophage clearance? Because that is really an important issue. And then the old problem for all of us that work in this field is that we all feel that it should be combination therapy. You can't convince any of the drug companies to do those type of studies. And I'll wind up by this, usually when you finish a talk, you show your laboratory, or you show your collaborators. I decided not to do that. This is because you need to realize that you have a lot of amyloid expertise here at Mount Sinai. And I said to Dr. Kohler before I started, the ligand part is extremely important. Mount Sinai Heart, starting with Valentin Fuster, who alerted me to Alzheimer's of the heart, which is really a very real entity, and a number of people, Sean Pinney, Martin Goldman, and Dr. Lalu Trinidad, and I've reproduced Martin twice. I guess there's some significance to that. And Radha Gopalan, very important for us. They're really doing some very interesting work, and I'll skip down somewhere to Maria Trivieri, who's doing some very nice imaging work. You have a very strong myeloma group, and it's getting stronger. And with the addition of Bart Barlogi, and so we have people that I can relate to, because I used to give the chemotherapy years ago. I wouldn't want to go near that now, given all the things that you can do. Our transplant program, I mentioned bone marrow, and Tom Shiano has been very important for the TTR patients and vetting the TTR patients. In nephrology, John Winston has interacted with me a lot on the AA amyloids. Neurology, David Simpson, who's the head of our unit. Jessica Robinson-Papp has collaborated with us on a number of our neuropathy studies. Bob Phelps and I just published a big review on cutaneous amyloid, or we're in the process. Peek Wu has been very important for ENT. You've got people in ophthalmology. I'd be remiss if I did not mention the Alzheimer's Disease Research Center. I was thinking on the way in, the first time I met Dr. Davis, he was giving a lecture out on Long Island, and he was talking about treatments for Alzheimer's disease. And he mentioned two drugs. One was Plaquenil and the other one was colchicine. And I remember going up to him and saying, "What are you doing? Those are our drugs. Why are you using them?" And so there's a lot of interest in this, and Sam Gandy, Nick Robakis, Dr. Goet, who's a recent recruit, very important, and it goes on. And then structural and chemical biology. Roman Osman has been is very interested in this area. Ram Wang has done a lot of the mass spec that we did. And in pathology, it's a growing group. And Dr. Babic, as I understand it, is actually trying to set up mass spectrometry. So there's a lot of expertise here, and I'm assuming that will keep us going for the next 15 years, which I guess will be the next time I'll come back and talk to you. Thank you.
Questions?
That was fabulous, Peter. The energy landscapes that you showed are really the key to all of this. So I was wondering, how good is the molecular dynamic simulations with any of these mutants in terms of looking really at the intermediates and the ability to predict the sand that you need to throw in to prevent the contacts that are made? We should be getting close to that, I would hope, by now.
I think the answer is yes. There is increasing technology that we can use, and more importantly, some very, very good people who have actually gotten interested in this. A fellow named Chris Dobson, who is at, I think, Cambridge, and David Eisenberg, who's in Los Angeles, have been very, very clever at looking at these structural modifications and developing in vitro systems that we can look at the specific interactions, as well as the transitions that occur as you go from monomer to these amorphous aggregates, and maybe through a different route to the oligomers.
And if you stop all new polymer, how variable is the dissolution of existing amyloid?
Well, that's really the whole problem, and that's one of the reasons why I think a lot of these early studies, which we have been involved in, are flawed. Because one of the things we used to joke about was that if you wanted to isolate amyloid, take a specimen and leave it on the bench and go back to it about 5 months later. Because what happens is everything gets digested except the amyloid. The clearance mechanisms that we have are really imperfect for this. And so we need something that would be, that would enhance the clearance once it's there. Now, other people would argue a little bit differently, because the argument, particularly in the Alzheimer community, is the amyloid really important? Is that really what causes the problem? And there's a lot of thought that that may not be the entire answer, that it has to do with the, well, in regard to AD, the co-deposition of the tau, or it could be these oligomers which are not going to be affected. So it's, you know, people are looking at each of these different components, and we're getting a lot better at it.
So I'm just curious, there's so many different types of amyloid that you pointed out. Is the mechanism of misfolding the same for the different types? Is there something in common where you could attack amyloid as such, forgetting what the 100 and some different types are?
Well, there are two different theories about this. The first theory says that there's a generic abnormality that has to do with this beta-pleated sheet configuration and the stacking of these. And that group basically looks at these X-ray diffractions, for example, and say, well, they're all the same. Doesn't make any difference what type of amyloid. But there are others that think that there are distinct differences between amyloids with regard to whether or not there are significant oligomers that are there. And also, the other thing that is much discussed is what are the kinetics of this? Because it's not just a matter of making the amyloid, it's a matter of how you nucleate the amyloid and what the nucleation part of it might be. That's very disputed. But also the other part about amyloid is that it tends to spread. And so the issue is what makes it elongate. And these may be very different depending on the particular types of amyloid. So you see a type of amyloid where you see a little circle in an organ, but you also see amyloids where it just seems to spread throughout the organ. And I didn't show you that, but that can happen with even the models we have.
Thank you very much.
Sure.
Please.